Thank you, everybody, for being here and listening to my presentation. Today, I'm going to talk about the phosphorus deficiency leads to stunting, but what does it really mean? I'm Marlon Retana. I'm going to start talking a little bit about my background. So I got a bachelor degree in agronomy from the University of Costa Rica. I worked with nutrition in seedlings in 12 horticultural species there. So when I finished my bachelor, I came here to US to study a master's degree in environmental horticulture in the University of Florida. I work with ginger and turmeric as alternative crops for Florida. Once I completed my master's, I started my PhD with here in the horticultural sciences in the University of Florida, working with Dr. Gerardo Nunes. And my research is related to phosphorus nutrition at different aspects: physiological, morphological, phenological, also yield responses related to phosphorus nutrition in blueberries. And why phosphorus is so important? Well, phosphorus after nitrogen is the most important nutrient in plants. In fact, the reason for that is that it's involved in many physiological, biochemical, and other cell signaling processes in plants. And the importance is because this is the primary constituent of critical biomolecules, such as ATP, NADPH, RNA, DNA, and sugar phosphates. So phosphorus is a key component for plants, for growth and development. And that's why phosphorus deficiency is one of the main factors limiting crop production in general. And the dynamics of phosphorus is really interesting in terms of related to the soil and with the fertilization. If we think of a root epidermal cell, this is a close-up of one small section of the roots. When we apply the fertilizer, the phosphorus fertilizer, as you can see here, unfortunately, only 15 to 20% of the phosphorus that we applied as a fertilizer ends up in the plant. And I have to mention that plants use phosphorus just in the form as the chemical form of phosphate, specifically orthophosphate. So phosphate enters the plant into a simple mechanism, but the reality is that most of the fertilizer ends up in the soil, fixed in the soil, because phosphate is really reactive to other nutrients. So let's say if the pH of the soil is low, the phosphate ends or it's bound to other nutrients like aluminum or iron. If the pH of the soil is high, usually it bonds with calcium. And also it could be bound with organic matter, forming organic phosphates. All these forms of phosphate are immobile and they are not available for plants to use. So we think of a plant that is growing in a phosphorus deprived, or in this condition of where phosphorus is fixed in the soil, plants have different strategies to deal with that, to overcome this situation. So like the classic responses to increase phosphorus bioavailability and also phosphorus use efficiency in plants are like modifications in the root architecture. As you can see in the image on the right, you see like this plant has a really large or big root system. And also these root systems produces several components like root exudates in order to make more soluble all these fixed forms of phosphate in the soil. And also it has some symbiotic association, especially with mycorrhizae in the soil. And also another strategy of the plants is the reallocation of photosynthesis in order to deal with the deficiencies. One of the most common enzymes that plants or roots release in a phosphorus deprived environment is the acid phosphatases. And these phosphatases, their function is to break the binds to in the organic phosphates to make more soluble the phosphorus in the soil. But also, roots produces low molecular weight organic acids, and the function is to release the phosphate from the other nutrients like calcium, aluminum, or iron. The thing is, all these strategies are really costly in terms of carbon and also nitrogen. So, like the plants in these conditions show different symptoms of phosphorus deficiency. And the most clear or evident symptom is the diminished growth, or what they call like a stunting, which is not grow at all. Should dry mass ratio, because of, like in this image, you can see that this plant has a really huge root system, but also the delay in flowering, and also ultimately they have reduced yield. And another key or clear symptom of phosphorus deficiency is like changes in leaf coloration. You notice, like, for example, reddish purple colors in the leaves because of the production of anthocyanin in the leaves in order to deal with the phosphorus deficiency. So with this complex situation about the phosphorus with the soil and the plants, we think of we're starting thinking about the deficiency in terms of growth reduction and also stunting. But what does stunting mean from a physiological standpoint? So what we try to answer is, what is the cost in the plants of the phosphorus deficient in terms of carbon? So our research objective is to determine the connection between phosphorus deficiency and carbon gain and loss in Southern Highbush blueberries. In order to accomplish this goal, we established an experiment. It was a hydroponic experiment in a greenhouse. I did it last summer from June to August, where I worked with 4 different of blueberry with different growth and also general architectural characteristics. So I started with the complete nutrient solution. Each plant grew in one container with nutrient solution. I have an acclimation of 5 weeks for all the plants with the full nutrient solution, which included 15 milligrams per liter of phosphorus in the solution. Once this acclimation period ended, I split the plants into 2 groups. One group continued to grow in a full nutrient solution with phosphorus, but the other half of the plants, they grew in a deprived nutrient solution with 0 phosphorus in the nutrient solution to see the responses in these conditions. I have to mention that I changed the nutrient solution every single week at the beginning of the week on Mondays. And I adjusted the pH every other day to make the value of the pH around 5.8 throughout the experiment. This picture, I have to mention that from now on, I want to call the plants that grew in a complete nutrient solution as plus P treatment, but the other is the ones with no phosphorus gonna be the treatment minus P. This is one of the pictures of the setup in the greenhouse that I had last summer. I provided with air pumps the oxygenation in each bottle throughout the experiment, which is really important in this kind of studies. And also I use aluminum foil, as you can see, in each bottle to avoid the light to enter the root system or the root area, and also to avoid the algae growth in each bottle. I measured several parameters in this experiment. I measured plant growth in general, like dry mass, phosphorus concentration, and phosphorus content. And I measured several root parameters like root architecture, the classification of fine roots, like hard roots, and other parameters related to roots. I also measure plant gas exchange, net photosynthesis, and root respiration. Also, I measure several parameters related to acid phosphatase activity in the rhizosphere and root carbon exudation as well. And finally, I measured the two most important indexes related to this phosphorus deficiency experiments, which are SPAT index and also anthocyanin reflectant index. But in this presentation today, I will focus the attention on the variables that are highlighted in red, which is related to the carbon use and for the rest of the talk. So how we did it? So if you see this plant, so we came up with a whole plant gas exchange system where we built a chamber to measure the entire gas exchange, the plant, like the photosynthesis or carbon assimilation in the whole plant with a chamber. But simultaneously, we built this small chamber for the root system where we measure the root respiration. I'm sorry. So in that case, we use one Cyrus for the net photosynthesis and the other Cyrus for the root respiration to measure simultaneously the entire gas exchange dynamics in each plant of the experiment. And this is I want to show here all the components of the system. You can see here in these two pictures the custom built cylindrical whole plant chamber. It is made of PVC, the wide one. But inside this PVC chamber, you will see in another picture later on the clear acrylic plexiglass chamber inside the PVC with a volume of 2.6 liters. You notice that I provided the light, artificial light, with this LED growth light strip that provided 256 photosynthetic photon flux density for each plant. And also to avoid to increase the heat in the system, I use this small fan here, and I put it on top of the chamber to provide like airflow, but not inside the chamber, but outside to reduce the heat in the system. And also I use this arm to attach the system in a bench in the greenhouse. I'll show you a picture later. And of course, I want to show you in the next picture, I use these air fittings here, as you can see, in order to connect the chamber to the Cyrus to measure photosynthesis. This is you can see the cylindrical chamber. I made sure that everything was really close with, I put a lot of silicone here to avoid to have leaks, air leaks in the system. Another important component of this system was the rubber stopper, as you can see here. With this rubber stopper, I was able to seal the chamber to avoid any air leaks when I measure the plants. And also you notice here like a small hole in the center of the rubber stopper. And also I created this space in order to open it and insert the main stem of the plants. And also when I close it with the plant, it created a really good seal to avoid any leaks in the system. Once we attach all the pieces or put all the pieces together, like the LED strip, like the fan, and also the rubber stopper, as you can see here, I made sure that inside this chamber, there were no leaks at all before I started the measurements. For the root respiration, we created this chamber with a opaque 2-liter plastic bottle, as you can see here in these pictures. And the same thing, I use these two air fittings here to connect these to the hoses to the Cyrus. And also I use the same rubber stoppers, as you can see here, the small hole in the middle to insert the main stem of the plants. And I was able to insert the whole root system to measure root respiration. I'm using I'm showing this image, the small image here, because in other experiments I use this. This is a 50 milliliter cylindrical chamber, and I was able to measure just like sections of the roots. If I was able to cut some pieces and measure the root respiration with this small chamber. But it's the same principle, right? Like the air fittings really create a really closed chamber in order to avoid leaks. That's really important in this type of studies. This is a video where I'm showing how I did it. I placed the entire root system in the root respiration system here. And in this case, I'm showing a bigger variety that I use, which is Colossus. I was not able to fit all the canopy inside the chamber. What I did here was to just insert one branch of the plant in the chamber, the photosynthesis chamber. But it was with the biggest variety that I work with. The rest of them, I'm gonna provide results from the two smaller varieties that I was able to enter the entire plant in this PVC chamber. This is the whole setup of the experiment, as you can see here, with all the plants one side and also the whole gas exchange system in one side. I measured the plants 3 times throughout the experiment, but I'm gonna show results just from the last evaluation at the end of the experiment. And also I have to mention that I measured the plants from 10, I try to do it from 10 a.m. to 2 p.m., right in the middle of the day during the summer. So how I got the information from these two sirens? Well, I use the CO2 differential measure automatically every minute for 15 minutes in each plant. Simultaneously measuring, I was measuring the root respiration and also the photosynthesis. I have some calculation that I use to get the results. The data I used was the highest value measure once they reach a plateau or a variation, as I'm showing here, the plateau. So for the big chamber, I needed like almost 15 minutes to circulate all the air inside the chamber to make more stable inside the chamber, to make the stomata really equilibrate with the air, with the environment, to get this plateau here. So it was 14 in the big chamber, 15 in the big chamber to get this plateau here. Once I got this plateau, the measurement that I got from this from the Cyrus was close to here, like it was the stabilization when they were stable to continue or to get the values here. What I did, I used this equation, the gas exchange for both for root respiration and also for photosynthesis, which is basically the difference between the reference, the air exiting the chamber, which is CO2A, minus the CO2 reference, which was 1400 micromoles per mole of CO2 entering the chamber. And also I use the chamber flow rate. In the case of the root respiration chamber, I use the flow rate of 150 milliliters per minute. But in the big one, the whole plant photosynthesis chamber, I use 400 milliliters per minute in the big one. And at the end, to calculate the milligrams the milligrams of CO2 per minute, I use the density of the CO2 here at SCTP, like the standard temperature and pressure. So that's the formula that I used in these calculations. But also I included in this work, in this research, the root carbon oxidation from the roots. So how I did it? Well, I determined indirectly by measuring exhaust concentration in the nutrient solution. What I followed was the Anthron method as per Landhauser and Sperling, which basically what it's consisted of I made a glucose standard curve of standard solution. And what I did was I changed the nutrient solution. It was the last week of the experiment. So I changed the nutrient solution the last week of the experiment on Monday. I collected samples in the same day when I changed with the fresh nutrient solution. And also I collected samples 24 hours after changing the nutrient solution to determine the carbon exudation in a period of 1 day or 24 hours. I collected some 20 milliliter samples in each bottle in all the bottles of the experiment. And then at the end, following this protocol, what I did was to measure the absorbance at this, at 620 nanometers in the spectrometer to determine the amount of carbon that were exuded by plants in 24 hours. With these three parameters, photosynthesis, root respiration, and root carbon exudation, I was able to determine the daily carbon gain of the plants. So basically, I integrated these three parameters, and I adapted this equation from the work from Nimali and Van Eersel. Well, I'm using the whole plant photosynthesis. I use the day length of the day in the summer, which was 13 hours of daylight, and I multiply by the time, 60 minutes per hour, right? So this was the whole plant photosynthesis in a day. But also I usually with this parameter, I subtracted the root respiration, the whole root respiration in a whole day. It is I have to mention the plants, they have root respiration throughout the day, 24 hours. So that's why I use 24 hours period here, and again multiply by the time, the minutes per hour, right? And also this is another way of loss of carbon, which is the daily whole root exudation per day. So with that, I made the carbon budget of these plants. Like I said, again, the whole plant photosynthesis from the CIRAS. I use the CIRAS-4 to measure the whole plant photosynthesis. I determine one of the carbon losses, root respiration, with the CIRAS-3, and also the root exudation from from the entire root system of the plants. So first of all, I want to share some of the results here. I'm going to start with the dry mass. As you can see in this graph, this is the dry mass at the end of the experiment. This is the picture of the last week of the plants in the last day of the experiment. You notice here the dry mass in grams, and also the different organs, roots, canes, mature leaves, and also young leaves. And you notice, I have to mention that in this presentation, I'm presenting results from the two smaller varieties that I work with, which are farthing and also key crisp. So you notice, I'm going to start talking first about farthing. As you can see here, these are the phosphorus deficient plants, the ones in the orange. And this is the phosphorus with plus P plants, which are the plants that grew with plenty of phosphorus in a nutrient solution. And you notice in all the organs, we had statistical differences in all the organs, where all the plants that grew in the deprived nutrient solution, they had lower dry mass. So we see here, like these plants, they stop growing, or they grew so slow compared to the plants that grew with the complete nutrient solution with phosphorus. In the key crisp variety, as you notice here, I didn't have any statistical differences. However, you may notice a tendency here where this plant, phosphorus deprived plants, they had higher root dry mass because of this, one of the symptoms of phosphorus deficiency, like I told you earlier, which is the production of really huge root systems. But also you notice this a decay in canes and mature leaves. So it is a tendency. So it's because this variety maybe it needed more time to get to this point of deficiency. So, but it's a tendency that these plants were they had lower biomass accumulation in the phosphorus deprived plants, except for the young leaves. You can see like it's quite similar, but it's because of the reallocation of carbon in these, specifically in canes and mature leaves, to roots and to young leaves. You can see the reallocation of resources. And again, you notice the reduced growth in farding plants. And we made sure, and we were able to induce phosphorus deficiency, which is important to continue this study, right? Because you notice here that in both cases, I'm going to walk you through these graphs. This is the phosphorus concentration in these scales. And this is, again, all the organs that you can see here. I divided the plants into different organs. And you can see here the two varieties that I work with. And in both cases, we succeed in taking these plants to deficiency, right? This line, this red dashed line is the threshold, the deficiency threshold for blueberries in terms of phosphorus deficiency, which is 0.12, the concentration of phosphorus. So you notice that these plants, in both cases, in farding and in key crisps, were under deficiency in this experiment. So, but the rest, like the other plants, like the plants with full nutrient solution, they were above this line in both cases, which means they were not deficient. So we were satisfied with this result, so we continue with the rest of the information. Once we knew that these plants were deficient, and this is a picture of the last day of the experiment. This is the farding plants, as you notice, the reduced growth. This is the minus P plant, as you can see here, compared to the plus P plant. And you may notice here the root growth as well. This is the phosphorus deprived plant had higher root system or more biomass in the root system, as you can see here. And this is the typical response, like reddish purple coloration, starting in the older leaves, but also they started to present the symptoms in the new leaves, as you can see the margins of these leaves, and of course is contrasting, right? The growth in both cases. In the key crisp, as you may notice, it's not a really contrasting difference, right? But it is a trend that these plants eventually were in a way of the nutrient deficiency or in the way of reduced growth. But one of the interesting situations with this variety was the differences in terms of root growth. You notice here the phosphorus deprived plant produces a really massive root system compared to the one with the full nutrient solution. And this picture is one of the leaves of this variety. And you notice that it started to produce visual symptoms in the leaves of phosphorus deficiency. Usually the symptoms started, like I said, in other leaves of the plants. And you notice that they started in the margins, this coloration. And this is because of the production of high anthocyanin concentration in the plants. With this information, that now we know that these plants stopped growing and these plants were affected because of the phosphorus deficiency, I'm going to start talking about the gas exchange results. So I'm presenting here, I'm going to start with the net photosynthesis. Based on the equations and the information that I provided earlier, I was able to determine the whole plant photosynthesis in a day. This is in micrograms of CO2 per minute. Sorry, this is not per day. I'm gonna show the daily results later. This is in a minute in these plants in the last week of the experiment. So you notice that we had differences in farding, as you can see here. I'm sorry. We had really huge differences in the whole plant photosynthesis. You notice that the phosphorus deprived plants, they had lower rates of photosynthesis, but no differences in key crisps. But there's like a tendency that they were reducing the photosynthesis at this point of the experiment. And in these two other graphs on the right, I'm showing the whole root respiration, again, per minute in micrograms of CO2. And you notice that in both cases, in the root respiration, we had different statistical differences in both cases. You notice higher root respiration in the plants, in the phosphorus-deprived plants in both cases, you see here. And in terms of numbers, farting minus P plants had up to 47% left carbon assimilation and almost up to 49% more root respiration than phosphorus, that plus P plants. And like I said, no differences in Q-crisp in photosynthesis, but it has a really big difference in terms of root respiration, basically because of the size of the roots that I showed you earlier. With this massive root system, they produce a really high root respiration in a phosphorus-deprived situation. The next variable that I'm showing is the root carbon exudation here. In both cases, you notice that phosphorus-deprived plants produce higher root carbon exudations. And it's quite similar proportion, 85 and 88% more carbon exudation. The difference is the magnitude, the scale. You notice that this is not the same scale, it's the double. This is 2.5 milligrams of carbon per day. The other one is 5, but you notice it's because of the size of the plant. So bigger plants produce more exudation, right? But it is interesting the difference compared to the plants that grew in a complete nutrient solution. So with this scenario here, up to this point, we were able to determine the carbon budget or the carbon cost in a daily basis with these three parameters: for photosynthesis, root respiration, and carbon exudation. So this is the budget or the carbon costs. So I calculated the daily carbon gain in milligrams of carbon per day. In both cases, you notice the differences. It's a really huge difference, right? In fact, it is phosphorus-deprived plants had 5 times less daily carbon gain compared to the plants that grew in a complete nutrient solution. And in the case of Q-crisp, the magnitude was smaller, but still high, right? Like 3 times, they had 3 times less carbon gain per day. So with this picture, with reduced photosynthesis that I showed you earlier, increasing root respiration and also increasing root carbon exudation, we have carbon deficit, right? So basically what we saw is that in a phosphorus deprived environment, these plants suffered a lot because of that situation, even though it was a short-term experiment, right? It was they grew for only 8 weeks with the treatments, right? With the phosphorus deprived nutrient solution, even though in 8 weeks we had the results. So that's the meaning of stunting, right? It's like in a phosphorus deprived situation, the plants had a lot of responses or also strategies in order to overcome this scenario. They are trying to survive, so they are able to reallocate carbon from canes or branches or stems to roots, and also from older leaves to new leaves. So it is the meaning of growth reduction at the end, right? And the cost in terms of carbon because of the phosphorus deficiency. So I want to finish my presentation with the summary. So basically, Southern Highbush Blueberry displays several strategies to overcome this phosphorus deficiency, like I said. The responses to phosphorus deficiency in Southern Highbush blueberries are genotype specific. So it's really interesting how, depending on the variety, the responses are different, right? The carbon budget and distribution in the plant play an important role in the responses to phosphorus deficiency, like I mentioned. And the stunting or reduced growth has to do with carbon imbalance in general, right, in phosphorus deficient plants. So I want to finish my presentation with saying uh, thanks to all the members of the small fruit horticulture lab where I work, especially to Dr. Gerardo Nunes, my advisor, and also our lab manager Dylan Kovac for all the help throughout this research. I have to say thanks to Florida Department of Agriculture and Consumer Services, specifically the Office of Agricultural Water Policy Award, to provide funding for this research. Also, the calls like the Dean's Fellowship that is giving me, like, providing funds to conduct this, to stay here doing this research, and of course PP Systems to provide the travel award for me to present this research. And I'm really happy because I'm able to present this research in the next American Society for Horticultural Science meeting in Hawaii in September. So I will present this research there. And thank you, PP System, for that. And thank you all for listening to this presentation. This is my information contact. If you want to, if you have any further questions, I'll be more than happy to answer. Thank you very much.
Oh, thank you, Marlon. You did an amazing job, and it's really interesting research, so I'm sure it'll be really well received at horticulture. Thank you for attending the webinar. Contact us today to learn more about the Cyrus 4 portable photosynthesis system, the 4th generation portable powerhouse elevating the high-level field research experience worldwide.